Good morning. It's nice to see some of you here this morning. How many of you leave town as soon as class is over? A fair number. The others are going to stay in town? Yes? Well, we wish you all a happy Thanksgiving. So let's begin then. And this morning we start with all things the rectum. So the end of our digestive tract, we followed this all the way down, and now we're just at the last portion, the rectum and the anus. The rectum will be straight, that's rectus, straight, a straight tube, in contrast to what we've seen for the colon, as it has its ascending, transverse, descending, sigmoid portions. It will have simple columnar epithelium, so what can you say about the digestive system from the esophagus to the rectum? What kind of epithelium do you have? Simple columnar throughout, right? Every part. Stomach had it, small intestine, large intestine, now the rectum has it. It doesn't have stratified squamous like the vagina. It has simple columnar epithelium. And it will have the smooth muscle walls, as you saw in the other part of the digestive system, except the colon where we had the Taenia coli. So we'll say there are no Taenia coli in the rectum. We have two layers, inner circular and outer longitudinal. So that you can have the strength in eliminating the feces, have the longitudinal muscle surrounding the need strength. In eliminating feces. And then we come down to the anus, the end of our digestive tract. And here we're going to have stratified squamous. And we'll have two layers of muscle forming sphincters. Let's just put two sphincters. Of muscle. The first will be smooth muscle, and the second will be skeletal muscle for voluntary control. So that gives us our very brief trip through the digestive system. But I do like to tell my little stories. Once I was called, you know, a little yellow school bus that goes through the body, that children, did you ever follow that little yellow school bus when you were little? Well, I was called and asked if I would consult for them from New York on the digestive system. And I said, sure. And so we started, and I said, well, we go down the esophagus, stomach, and I was just describing it to them briefly. We get to the large intestine, we go to the rectum. We can't do that on television. So did anybody ever see what happened to the little yellow school bus in the digestive system on TV? They get it down to the beginning of the large intestine, and the child has a big hiccup, and the little bus comes out the mouth. So I hope when you go into the media that you give the facts and let people get adjusted. They've got an anus, they've got a rectum, we've got it all. Everybody has it. Why are they so hung up? Isn't that strange? Boy, the thought of it. All right, so let's go on now. And I'm putting the tongue in next because we are coming back to the mouth to do the salivary glands. And I just wanted to review the innervation of the tongue with you, because very few structures have the amount of innervation that both the eye and the tongue have. We said, how many of our cranial nerves innervate the eye? 6. And now how many innervate the tongue? So let's just review. Tongue innervation. We're only doing this because we're coming back up to the mouth for the salivary glands, as I said. So what's general sensory to the tongue? It's good we had a review. 5, yes. General sensory. Remember, 5 took care of nasal cavity and oral cavity, and the general sensory to the face will be 5. So what is motor to the tongue? Everybody knows that one. 12. Never forget it. That's the last cranial nerve. Remember the story I told you, how the lady saved the autistic girl's life by knowing her cranial nerves? Stick out your tongue. She knew she came that far down the brain stem, she'd be at the end of the brain stem, then she could work back up, and saved the little girl's life from being autistic. She said, if you understand me, stick out your tongue, and she did. And she hadn't spoken in 12 years. That's why we drill and drill and drill. You never know when you can use this information. So this is 12. What supplies the anterior 2/3 of the tongue's taste buds? That one you do know. So that's 7. How about the posterior 1/3 of the tongue's taste buds? What supplies that? 9. How many of you have had a thorough neurological exam? Anybody? 1. Did they test your taste buds? Yes. Could you answer, or was that before you had it? No, you could answer. Good for you. Because they're testing if your 7th sensory nerve is intact. I mean, we said what we did for motor 7, but when you get a thorough physical for neurology, you go through all your cranial nerves. So I want you to be knowledgeable, and so you know that there are 4 of the 12 cranial nerves just to your tongue. All right, with that, let's move on to our salivary glands. We have 3 pairs of salivary glands. So we have the parotid. We'll take each one separately. The submaxillary and the sublingual. So why do we have our salivary glands? They will then produce and discharge secretions into oral cavity. And why? What's the purpose? Very simple, straightforward, to moisten the food, to ease with swallowing. To moisten the mouth, let's put oral cavity. To assist with speaking, we all know when we're scared and we have no saliva, it's hard to speak. So moisten the oral cavity, assist with speaking. And to add enzymes to the saliva. What's one of the examples of an enzyme in your saliva? Amylase, everybody knows that one. Amylase acts on carbohydrates to begin breakdown. How many have ever played on the DNA molecule at the Lawrence Hall of Science? Do you know what that DNA molecule represents? Amylase. We thought it would be fun to have a DNA molecule of spit for the kids to play on. So when they see it in their textbooks when they're in college, they won't be afraid of DNA. We've played on it. That's why the big molecule is up there. All right, we also have glucagon. Glucagon will raise blood sugar. So what is the location of these glands so they can secrete saliva? Let's take parotid first. It's the largest. What does para mean? Near, next to. Let's just say near. And otid, what's that going to be? Guess. Ear. So it's near the ear. It's going to be anterior, inferior to your external auditory meatus. It's anterior, inferior to your external, remember it, auditory meatus. Does that mean now that it's going to be anterior, posterior to sternocleidomastoid? I'll get your sternocleidomastoid. Find out where it is as it goes up there. You've got a gland that's going to be anterior, inferior to your external auditory meatus. And my question is, you've got a sternocleidomastoid coming from the sternoclavicle up to the mastoid process. Where's the mastoid process? Behind. So is my parotid anterior or posterior to the sternocleidomastoid? Anterior, sure. I want you to work these out. I want you to have the knowledge to play with it. So this is then anterior to sternocleidomastoid. Students are always asking, well, will our next exam, cover the whole class? Well, sure, this is part of that you've learned, but it's also being brought in again in a different context here, right? You know your sternocleidomastoid. You should, right? All right, so this gives us our parotid, and now it's producing secretions. Where are those secretions destined? to the oral cavity, but where? Do you know where the duct enters the oral cavity? The duct for the parotid enters opposite the second molar of the upper jaw. So you with your tongue, you can count over, you can feel your canines, right? And get to the second molar, not premolar, second molar. So go 1, 2, next one, then you know your parotid duct is going to come in there. When you get your teeth cleaned, do you notice sometimes when they're cleaning your teeth that they spend a little more time by that second molar scraping? Because frequently you form more tartar there as this duct is pushing in the saliva. You don't notice that? I used to notice that. No? All right. So opposite the second molar. That's the second molar, upper jaw. Now we have 2 other glands. The submaxillary, it's said to be walnut size, so most of us know the size of a walnut, so you get a visual image of the submaxillary. This is our second one, submaxillary. And we said walnut size. Where is it? It's going to be just inferior. Did I say maxillary? I should say mandibular. I'm sorry. I get so excited with all these. But it has to be mandibular because of its location, because it's submandibular. Submandibular, I'm sorry. It is the inferior to the body of the mandible. And then we have the smallest one, which is the sublingual, so you know where it is. It will be in the floor of the mouth. And these 2, 1 and 2 ducts combine, 2 and 3 ducts, 2 and 3 ducts join, and they will enter the floor of the mouth lateral to the frenulum. How many know what your frenulum is? You know, when you look at the inferior surface of your tongue, you see this mucous membrane attaching the tongue to the floor of your mouth. Officially, that's your frenulum. Prevents you from swallowing your tongue. Your tongue's attached, right, with the frenulum. So lateral to the frenulum. And the frenulum then is the mucous membrane that attaches tongue to floor of mouth. Have any of you ever seen your saliva when you have done something in your mouth that causes it to eject? You're reading. You've never seen that? We usually have students who've had that happen. You've never had it happen? Sure, some of you are nodding. I mean, I have, so I know it happens, but the books tell you it happens. Sometimes if you put lemon or something really very tart, that it constricts the walls of the ducts, and they eject saliva. And gets on your page. It's rather messy, but it's kind of interesting. It lets you know that you have ducts coming in to the floor of your mouth, all right? You might try it on each other. We'll see. If it doesn't work, don't blame me. It doesn't have a system that's strong enough. Anyhow, let's go on then. That tells us a little about our... One thing we could say of how much saliva is being formed each day, since I like to put these in to give the dynamics of one system affecting another, from 700 to 1,000 cc's of saliva is formed each day. So again, it lets you know the importance of having a healthy colon that's going to be absorbing all this fluid that's being produced. Now then, let's move on to another gland. Let's take the liver. The liver is the largest gland in your body. How much does it weigh? 3 pounds, right, good for you, you remembered. What other major organ in your body weighs 3 pounds? Your brain, right. So now where do we find the liver? We find it inferior to the diaphragm. We know that represents diaphragm. And we'll have 4 lobes. We'll have right, left, quadrate, quadrate, and caudate, caudate. So you can tell roughly where they'll be. The largest lobe is over in our right abdominal upper quadrant. Large lobe, this will be right, and then we'll have left over here, and there's a membrane between the two. It's called a ligament, the falciform ligament. Separates the two. And if we look on the inferior surface of our right lobe, we'll find the 2 smaller lobes, posterior. Which one do you think is going to be posterior? The quadrate or the caudate? Caudate, good for you. Caudate. So anterior, inferior right lobe will be the quadrate. So now, the liver is a very, very richly supplied with blood organ. Rich blood supply. How much it bleeds when it's injured. So, review. Where's the arterial blood coming from for the liver? I'll give you a hint. That artery is coming off of the celiac. Where's the celiac coming from? Abdominal aorta, remember, 3 branches of the abdominal aorta, celiac, superior mesenteric, inferior mesenteric. We've got the celiac, and it's giving off an artery that goes to the liver. What would you call it? Hepatic artery, sure. Hepatic artery. But we also have a major blood supply coming in. What's the second one? The hepatic portal vein, right. hepatic, good for you, portal vein. I had a student come back this morning from Pittsburgh Medical School who took our class. She said she just loves medical school because she has this grounding. She knows it, and she just can't think of anything she'd rather be doing with her life than back there in medical school, building on this material. Anyhow, she came all the way to say hello. Hepatic portal, where is the blood coming from into the hepatic portal vein? Well, it's bringing nutrients, so it's coming from the stomach, the small intestine, pancreas, and spleen. Because it's a vein. It's not carrying arterial blood. It's carrying venous blood rich in nutrients from stomach, small intestine, spleen, pancreas. So, with this buildup now, let's look at the histology of the liver. Histology of liver. Does anybody know the structural and functional unit of the liver? It's the liver lobule. What's the structural and functional unit of compact bone? Aversion system. What's the structural and functional unit of skeletal muscle? Sarcomere. Sure, you've got it there, but it's nice to bring it out and use it. So now we've got the structural and functional unit of liver is the lobule. Liver lobule. So we're going to design a liver lobule for you then, so you can see that, how it functions. The main cell here is going to be the liver cell, the hepatocyte. Hepatocyte. The hepatocyte is to the liver, the neuron is to the nervous system. And it's going to be arranged in this lobule, which is a six-sided figure. This is my lobule. And it will have in the center a central vein. Central vein. And just roughly, we'll put the basic arrangement of these liver cells. They say they're in plates, sort of radiating in this way. I've left great spaces in between which aren't there, but for giving us our purposes, these represent plates of hepatocytes. And so now we're bringing in arterial blood and we're bringing in our portal blood. So our hepatic artery with O2 blood and our hepatic portal vein with nutrients. Will be found in what are called triads. What's a triad? At the point, we will have hepatic artery, hepatic portal vein, and a branch of the bile duct. The next one, hepatic artery, hepatic portal vein, and a branch of the bile duct. Because then the hepatic artery and vein here can liberate their contents to flow between the hepatocytes into the central vein. The channel by which they're flowing is called a sinusoid. So a sinusoid is between here it's a modified capillary. It will have phagocytic cells sitting within it. We'll put a few little phagocytic cells in here. Does anybody know what we call the phagocytic cells of the liver? Phagocytic cell. It's called a Kupffer cell. Kupffer cell. What's an important structure that it's phagocytizing? Old RBCs, old worn out. The liver destroys the old worn out. So phagocytizes worn out RBCs. So this gives the oxygenated blood and the nutrients to the cells and then goes into the central vein. Where's the central vein going? Central vein empties into the hepatic vein, and the hepatic vein empties into the main vein in the abdominal cavity, which is the what? Gracious. Inferior vena cava, sure. Inferior vena cava. Very, very basic. Now we've got to worry about what's coming into that bile duct. And in order to do that, I want to make another drawing which will elaborate on the hepatocyte and its function. Let's start with, here's our sinusoid. For blood, and we'll have our hepatocytes forming a wall. These are hepatocytes. And now we'll have the other side of the cell. So we've made another channel. This channel will be a bile capillary. These are my hepatocytes again. Same hepatocytes that were taking substances from the blood. But in my bile capillary, the hepatocyte is producing bile. So the bile will be going down my bile capillary and coming to my bile ducts. Now, isn't that a remarkable cell? That it can be producing bile, absorbing blood, have two channels for it. Now this bile that's coming off here, what does bile do? We talked about it before. Emulsifies fats. So it's got to get down to the duodenum. And I'm not going to give you all the series of bile ducts. You can get that in your advanced courses. But it will go down through bile ducts, which one will lead to the gallbladder. For storage of bile. And the other duct will go to the small intestine, to the duodenum. To duodenum, where it will liberate the bile into the lumen. Now, one thing, where is this gallbladder? It's on the inferior surface of the right lobe of the liver. So we had if we had our liver and we had our right lobe, which is most, we'd see peeking out on the inferior surface the gallbladder. So the gallbladder on inferior surface of right lobe of liver. So what tells the gallbladder when to contract when you've got fat in your duodenum? So let's say we get fat in duodenum. That liberates an enzyme enzyme name? Cholecystokinin. Cholecystokinin. Should be up there. Guess it's not now. Cholecysto, C-Y-S-T-O-K-I-N-I-N. And cholecystokinin then acts on the gallbladder. Telling it to contract. And it contracts, produces bile that goes into the duodenum and begins to emulsify that fat. Isn't that a nice system? Like a circular pattern there. I once sat next to a man on an airplane and we were stuck for an hour or so for some reason. And I asked him what he did. He said he was a lawyer. I said, oh, he said, but what I'd rather do is study the liver. He didn't know me from Adam. He didn't know I knew anything about liver, but I just sort of looked at him and let you know that he recognizes what a phenomenal organ this is. So as we look at the functions of this liver, one type of cell, let's see what it can do. We've already shown that it can absorb blood, it can put out an enzyme, but let's look at the functions of the liver. Have you ever thought about your liver before? How many eat liver? It's good, isn't it? Right. All right, very healthy, and you'll see why. Functions of liver. So let's first look at its dietary functions. Well, we've just given one, and that's to form bile to emulsify fats, so we can break down the fat for digestion, right? So form bile to emulsify fats. For digestion. What does emulsify mean? Break down. You know that if you get some butter and you put it all over your hands and you put it under the faucet, nothing happens, right? You've got fat there that's not broken down. Then you get a little bit of soap, and you put some soap in there, that breaks up the molecule, you put it on the water and it washes away. Well, that soap is essentially representing what bile does. It breaks up the fat so that it can be utilized. So we're forming bile to emulsify fats. Then, what have we got? It stores glycogen. It stores glycogen. Same cells. And glycogen then is a source of glucose, right? What does the brain burn? Where does the brain get its energy? From glucose. So glycogen can turn into glucose. We'll just put to feed the brain. So what happens if you become an alcoholic and you destroy these hepatocytes? This is severe alcoholism, not gentle. So you destroy hepatocytes, replace them with connective tissue. So you have no glucose for the brain. So you have a demented brain. So when people always ask, well, what's the effect of alcohol on the brain? It's more serious. What's the effect of the alcohol on the liver? Knocking it out first. And it does affect the brain too. But here you've knocked out the liver. All right? The hepatocyte stores vitamins. It stores vitamin A, vitamin D, vitamin E, vitamin K, and B12 as examples. So we're always told to eat our liver to get the vitamin A. It's very rich in these vitamins. Now, what else does it do? It will modify, let's modify carbohydrates, fats, and proteins for utilization. So now what are other functions of the liver that aren't necessarily directly related to digestion? Still, these are just little, here's your little hepatocyte up here. With all its functions. So, other functions, let's call it. What else do we think it can do? I'm sorry? It will, in the fetus, form RBCs. You know, your RBCs are forming where? In your bone marrow. When you were a fetus, in fetus, liver forms RBCs. What do we call the formation of RBCs? Erythropoiesis. Good. Begin to use the language. Again, thank you. We have mentioned previously plasma proteins, and we said certain plasma proteins were formed in the liver. So we have fibrinogen, and prothrombin. These are for blood coagulation. And then we said just a moment ago that we have macrophages called Kupffer cells. Macrophage. They destroy old RBCs. So how old is an old RBC? Anywhere over 120 days, right? So when they ask you how old you are, you can ask, how you mean my RBCs or my lining cells for my small intestine? Do you remember how old they are? 4 days, sure. All right, now what else do we have here? We're going to detoxify nitrogenous wastes. Detoxify nitrogenous wastes, and then produce urea and ammonia. One little cell, little type of cell. How can it possibly do all this? Isn't that amazing? So when you think of eating your Thanksgiving dinner, what's happening to your liver and all it's doing for you, I'm sure the other guests at your dinner table will be interested.